Okay, thank you, Aaron. So thanks for the invitation. Thanks all of you for coming to here. I hope to participate a little bit, so bear with me. I'm going to try a couple new things with this talk, so hopefully the technology will work for me. So Aaron asked me, to talk about soil health, which is a really broad topic. My personal sort of research expertise is more in the biological component of soil health, so that's going to be my main focus, but obviously soils are integrated systems, so we can't really tease one thing apart from anything else.
Hey, Rick?
Yep. Can you guys hear me in the back? Is that better? Okay, so I figured we'd start trying to get everybody on the same page, right? So what makes a soil healthy? It's a question I have been asked and that I have asked for years now and still struggle a little bit with giving an answer to it because it is so broad. So we have, of course, the National Resources Conservation Service definition that you're probably very familiar with. It's not the only definition out there. But the key thing to see here is that these definitions are really vague, right? They sort of say a healthy soil is one that continues to do the things that we want soil to do. So that doesn't tell us a lot specifically about what makes a soil healthy, what makes a soil unhealthy. What particular things should we try to change or maximize to make a soil healthier? So that's led me to come to this conclusion over the years that soil health is really up to us to define, us being anyone who cares about soil, whose living depends on soil, right? Which is really all of us because we all eat. So I thought a good place to start off is to get a little feedback from the room on what, more operationally, sort of what do you want your soil to do for you? Or what do you want your soil to do better than it currently is? So the way this will work, there's a little website there. If you have a smartphone or a tablet or laptop and you can get on the web, if you can go to that website, it should open up sort of this question screen, and you'll have an option to enter a code number. So the code number is there. And there's a question. It's just this question again. And you can sort of type, whatever short little answer you want. I think you can enter multiple things. Oh, is it? There we go. Is that working for you now, Leah? It should be. It's up there in the blue box. Okay, I'll let this answer cycle in for a while and see if there's any consensus here. So as these keep coming in, I thought I'd highlight a couple of things you've picked up on that are similar to what I've thought about, right? So there are kind of a couple classes of answers here that are answers that are quite, I would say, kind of operational, right? Like make us profitable. We want systems where it's sustainable in the economic sense to keep growing the way we grow. But you also see answers that are more kind of biological, right? That's centered around the plant. Is it feeding the plants? Is it growing great crops, nourishing crops? So again, that's a practical definition that has an outcome, right? You know if your soil is healthy because your plants are doing well in it, but you don't know why. And then you have other answers that are getting more specific to particular aspects of soil function, right? Producing more organic matter or holding on to more organic matter and holding on to water, being resilient to climate variability, so resilient to too much rain or not enough rain. So that's what I want to keep in mind as we talk through the rest of this, and if you think about soil health, is that let's keep all three of those ideas in mind, that we want soil that produces an outcome that we want, right? Which are productive, healthy plants. That does it in an economically sustainable way. So, right, we want our soil's got to be working for us both sort of in a biological sense and operational economic sense. And then we'll think about how do we, what specific things about soil do we want to focus on to achieve those goals? Let's see if I can juggle back to this. Take over, sweet. So we want to do soil as we just talked about. I'd like to break this out and think about scales. So in some of those answers that people were giving, a lot of them were thinking about this kind of local scale, right? So in one field, what does the soil need to do in that field? So we mentioned things like water holding capacity, providing fertility to our plants, suppressing diseases. Those are all sort of local scale benefits. There are also larger scale, regional, global scale benefits. So reducing soil erosion, reducing nutrient runoff, right? Eutrophication, big issue, at least in the Midwest here. We want those nutrients to stay on our fields, not in our waterways. Storing carbon, that's something that would benefit everyone on the planet if we did. So you can think about the benefits of soil health at multiple spatial scales. You can also think about temporal scales, and this came up in your answers as well. What's it doing for me this year, next year? So these, some of these benefits like reducing disease or increasing fertility, you might see those in any given year. You might think about these sort of medium-term benefits, right? I need a field that's going to work for me throughout my rotation cycle. It's got to work for different crops. You're trying to build that soil structure over the medium term. And then some of your answers were really long-term. Someone mentioned like 1,000 years. This soil needs to continue to be productive for 1,000 years. So how do you manage a soil in a way that you're getting those short-term benefits, but you're not sacrificing the long-term benefits? You're leaving something still useful behind for the next generations. Not an easy task, I think. So, all right, we'll try this again. Hopefully I'm getting a little more savvy with this. When Aaron asked me to do this, I thought, there are a million things I could talk about. So I made a PowerPoint that would probably take me 2 hours to get through. We don't have 2 hours. So here are all the things I am prepared to talk about, and I've got some backing in. And I'll open it up to you all to guide me a little bit, and I'll skip around to what you want to hear the most about. So hopefully this is still working. Advance the slide. Okay. So this should be a ranking. It's same code. So if you go to that website again, it should be under the same code. And you can rank these 4 topics. And we'll give people a couple minutes to do that, and then I'll proceed from there. There we go. Maybe I should hide this so you guys don't see each other's answers.
Rick, you're really taking a risk here.
I'm prepared to talk about all 4. Some of them I have more than others, I will say. So we might be able to get through 3 of them, depending on. Okay, it looks like consensus is emerging. I don't think too much. Top 2 topics. That's fine. That first one I actually don't have a whole lot on that one, so that will be relatively quick. I mean, I could talk for like 5 hours about my research, but I didn't. Figure you guys would want to hear about it. But we'll start there. I'm going to save this second one for the end because it kind of builds on some topics I talked about before, but I will be sure to leave time for it. Okay. All right, so let's, I'll skim through this indicator stuff. There is a couple of things I want to make a point on, even though I want the top choice. because it's relevant to the other things, and we'll move on to making a lot of time for those last 2 points. Okay, so how do we know if a soil is healthy? You're probably familiar with a lot of these. We've got indicators of physical soil properties, probably old hat, indicators of chemical soil properties. Good thing about those first two is that we know quite a bit about them. We know how they relate to crop yield. There's a good testing infrastructure. Biological soil health indicators. How do we know if our soil is getting better biologically? That is a tough one and an area of a lot of current research and a lot of active movement both in the research space and the commercial space. These first four up there, I don't know if how many folks are familiar with these. These are in the sort of the newer soil health test packages. Those first four really are chemical measures, right? These are all about chemistry. They're not actually measuring anything biological, but they're related to biology. Those last four are directly measuring living things. Let's skip this, that'll take some time. I want to talk a little bit about actually the top one on that list and the bottom one on that list as an example of organic matter. Everybody measures organic matter, right? If you're going to talk about soil health, that's probably the first thing people think about is, I want to have more organic matter. And so that's good to dive into just because that's actually an incredibly complicated phenomenon. Like, what is soil organic matter? It's not one thing, it's millions of things, really. And so just to give a little bit more background on how sort of this emerging understanding of soil organic matter among soil biologists. One thing is that we're paying a lot more attention now to different pools of organic material in soil. In particular, there's sort of this dividing line between organic material that is available for microbes to use, to be turned over, to produce, to mineralize resources for plants, that has what we call a short residence time, meaning that a piece of organic material, like off a plant, a piece of dead plant, enters the soil. It's going to stay in this pool for a short amount of time, months to years, maybe. So this is the primary food for most microbes. This is the stuff that's easily lost to disturbance. So when you till a soil, this is the stuff that goes away because it's so available to the microbes. If you have ever gotten a soil health test that presents this active carbon pool, which is actually a chemical digestion, permanganate oxidizable carbon, but the point is that's supposed to measure or at least correlate with this particular organic matter pool. So that's what you're looking at with active carbon. On the other hand, this other pool of mineral-associated organic matter. So these are chemical compounds that have actually been processed a lot in the soils. They've gone through the soil food web. That little bit of leaf has been chewed up by a microbe, broken down. That microbe may have been eaten by another microbe. Eventually that microbe dies. All along, the carbon molecule is sort of transforming. And then eventually it gets in this much more simplified state where it can get glued on to a mineral soil particle. So we call it mineral associated. This is really important because this is the long-term storage of carbon in soil. This is a slow pool. A carbon molecule that gets into this part of your soil can stay there for thousands of years because they're not available to microbes. They're protected by this mineral association. So if you ever start to think about carbon sequestration, and this idea of carbon banking, and we should pay farmers to bank carbon to mitigate climate change. If that's going to be at all effective, we got to get carbon into this pool. This is the sequestered carbon that's going to stay there. The carbon that's in that particular organic pool is, it's in and out, right? So having it in there 1 year doesn't tell us much about whether it's going to be there next year. And so just bring this up because if your goal is to sort of push carbon into the particulate organic pool, because that's the more active carbon that's feeding microbes, that's cycling nutrients, versus if you want to push carbon into this slow mineral-associated pool, you need to do probably different things. So it's not, there are some trade-offs sometimes with soil management, right? So that particulate pool, you need to maximize your plant residues, how much plant residue material is going back into your soil. That's where that pool comes from. Obviously, reducing disturbance, huge for this. This is a pool that's incredibly sensitive. The good thing about this pool is that there's no inherent limit to how big it can get. You can just keep pumping organic material into your soil, and this particulate pool will keep growing. On the other hand, this slow mineral-associated pool, to get carbon in there, you've got to focus on the microbes, because this is the carbon that's gone through the microbial part of the food web. So you've got to focus on microbial biomass. And sort of somewhat counterintuitively, to get the carbon in there, you've got to feed the microbes things that are easy for them to eat. Sort of like if you ate a pound of brownies versus a pound of broccoli, in which situation is your body going to get bigger, right? Same thing with microbes. If you give them easy to digest stuff like clover, more of that will end up in the microbial body. If you give them something that's hard for them to eat, like a log, wood or something, more of that is going to end up respired into the air and not end up in this mineral pool. Emphasizing deep-rooted plants, getting that carbon down deep where it's not going to be respired. Problem with MAM a little bit is that how big can this pool get depends on the soil you're dealing with, especially it's those minerals, right? So this can be inherently limited by your soil mineralogy. There's going to be a cap to how big this pool can grow in any given soil. Okay, so that's my spiel about organic matter. Quick spiel about, on the opposite end, everyone thinks about organic matter. This is an indicator that I've heard more about recently, this idea of looking directly at the microbes and at this ratio of fungal to bacterial components of the community. And so I thought I'd speak a little bit about this. This may be newer to some of you. So you'll hear about this a lot in my talk. A gram of soil can have a bunch of stuff in it. 100 million to a billion microbial cells, but hundreds to thousands of different kinds of microbes. So thousands of different kinds of bacteria, tens to hundreds of different kinds of fungi. And so, not surprisingly, in that big list of microbial species, you've got a lot of different roles that they can play. Some desirable and some less desirable, like the plant pathogens. And so one of the ideas with this fungal bacterial ratio is that you should aim to increase it, I think the way it's been marketed. But keep in mind that in both the bacteria and the fungal group, you have ecological roles that you want filled and ecological roles that you want reduced. So just collapsing that into these two numbers is pretty overly simplifying. And I just want to make a point. So when I was looking into this a little bit, like where did this idea come from that you should want more fungi than bacteria? It comes from an old idea in sort of natural systems ecology that there's a successional pattern that early in succession you have bacterial-dominated communities. As you go later in succession, you get fungal-dominated communities. That was the idea that was out there. But that's a bit of a misnomer. What really is true is that communities that are dominated by trees, by woody plants, tend to have a lot more fungi in them. And that is because of the way trees interact with particular symbiotic fungi. They have a kind of symbiosis, this ectomycorrhizal relationship, that no herbaceous plant has. That tends to produce a lot of fungal biomass. The other thing they do is that they produce wood, and fungi are really good at decomposing wood, or at least certain fungi are. Really no bacteria are. So if you have a community that's full of trees producing a lot of wood, you're going to get a lot of fungi because they're the only ones who can tolerate that. That should not mean, or should not be interpreted to mean that a forest soil is in any way healthier than a tall grass prairie soil, right? Or a tundra soil. They have a different, the prairie soils can be much more bacterial dominated because that's the appropriate system, appropriate community for that kind of system. Right? So don't be misled into thinking just because, forests have this kind of soil, that's what you want to replicate in a grain-producing field. That said, fungi are great. I would never encourage you to not like your fungi. They have a lot of unique properties that bacteria can't do. They can decompose with difficult substrates. They form those mycorrhizal symbioses, some of them. And in general, their kind of stringy lifestyle, they grow with these long, thin strings, is great for holding soil together. So they have these consequences on soil aggregation that bacteria don't. But because of that same morphology, they're really sensitive to disturbance. So if you're thinking about your fungal bacterial ratio as a proxy for how disturbed is my soil, it can work that way. because the fungi are going to be affected much more strongly than the bacteria are as a group. But I would encourage you, if you're going down this path, think about your total biomass pools in each of those. How much fungi do I have? How much bacteria I have? That's more important than the ratio of the two. So that little cartoon example there, if I had to choose, so the bar on your left would have a lower fungal bacterial ratio, right? It's only a third of the community is fungal. But I would much rather have that in my soil than the bar on the right, right? There's no reason to get rid of bacteria. You want to maximize your fungi, but also maximize your bacteria. Just maximize your microbial biomass. That's more important.
If you're shifting gears, Rick, I want to give people a chance to ask questions
Sure.
about this. Are there?
Yeah.
In regards to the depth of fungi,
Hannah's grabbing a mic. Hannah's a triathlete, so she can do that.
good job. In regards to the depth of fungi and disturbance, can you talk a little bit about that? And can they grow below that zone, and how does that interact with everything?
Yeah, one thing you will see if you have a relatively undisturbed soil, so you haven't tilled in a while and you start to build up some kind of a soil horizon, right, where there's sort of a layer of plant residue up top. Fungi are a little bit better than bacteria at sort of growing up into that. So in that sort of duff layer of decomposing crop residue, you're going to have more fungi than you will bacteria. As you go deeper into the soil, that'll shift. You also get a shift in which kind of fungi, right? So the fungi that make their living by decomposing plant materials need to be up high where the plant materials are. As you go deeper into the soil, you're going to get a shift toward those fungi that are making a living as symbionts on plant roots, because that's where their carbon food source is coming from. So, and I'm trying to go back to the question. If you mix that up, right, if you till the soil and sort of mix that organic matter in deeper into the soil, you sort of destroy that partitioning. And just a general rules in ecology is the more distinct niches you can have in a community, the more different kinds of species you'll be able to support, and overall, the sort of greater the functioning of that community. So if you, one issue with soil disturbance is that homogenization of like taking an ecosystem, a habitat that has all of these distinct niches in it where different microbes can make a living. And if you mix it up, then the microbes can't really separate out and have sort of, I'm a specialist here, I'm a specialist there. You'll end up getting a less diverse community because there's sort of fewer species that can dominate that general backing. If that helps. You see that?
Have you been studying this in either agroforestry systems or savanna pasture systems?
I haven't specifically, but other folks have for sure. And when you have that mix of trees and herbaceous plants, right, like you have in a savanna or an agroforestry system, if you can make it work, you'll get a little of both, right? Because there'll be fungi that are specialized on interacting with the grasses and fungi that are specialized in interacting with the trees. So like the diversity of the whole system is higher. You'll be in between effectively. You kind of end up in between that conifer forest situation, which is extremely fungal dominated, and the prairie that's extremely bacterial dominated. But it will also be patchy because you'll get little islands of fungi around the trees, especially if they form those ectomycorrhizal. If it's oaks or hickories that are part of your savanna, which a lot of times it is, they form the ectomycorrhizal symbiosis, so they'll have a lot of those fungi and sort of a little island around them.
Wouldn't prairies and things, wouldn't the plant exudates produce the bacteria more so than, that's not going to be fungal food, the exudates from the photosynthesis, right?
It will be too. Fungi are definitely not opposed to eating a juicy carbon molecule exuded by a plant. But you will get less, the reason partly, right, that you get more bacterial dominance in prairie systems is because the ratio of, what's your average microbe eating? Are they eating the carbon exudates from a root? Are they eating sort of root cells? Are they making a living off of this really complex carbon that's coming from leaf and sort of wood tissue? And so in a forest, there's a bigger emphasis on that complex carbon. And in the prairie, there's less of that complex carbon. So you don't need to have all those specialized machinery to eat it. And so the bacteria do relatively better.
Yes, you made the comment that tillage is hard on these things. And of course, in this room, there's lots of forms of tillage.
Right.
So we've switched to like a shallow tillage, high speed, this and that. We're thinking of going back to the plow because of the damn grass. So what's going to be harder on my microbes and all the living things, so I'm just blowing through them? Is the plow going to be worse than the shallow tillage? Several passes or one bad pass?
No, like Aaron's right next to you. You should probably answer that better than me.
Oh, yeah, that's you, Rick.
I know a lot about fungi, but Aaron knows a lot more about tillers. You know, the shallower you go, I think the better. In that you can, microbes, there definitely is a really strong gradient in microbial biomass as you go from the top of the soil down. So most of the activity is in that shallow layer of soil. That's where most of the plant roots are. That's where most of the organic matter is. Fortunately, that's also where the weeds are. I think in general, shallow is better than deep. But unfortunately, even a shallow tillage is going to hit the majority of the microbial community, especially those. The reason that fungi are so sensitive to tillage is because they grow in these like nets, right? So their hyphae are these really long, skinny things, little strings, and they grow in these elaborate webs in the soil. But that web is really concentrated on that top 5 to 10 inches, not even 10 inches probably. I mean, it depends on your soil type. It depends on the crop you're growing. If you have deeper rooted crops, the microbes will follow the roots down. So the more deep-rooted crops you can grow, the more carbon you'll be pumping in deeper into the soil, so there'll be more microbial activity deeper in the soil that would survive this shallow tillage and then be a resource to recolonize that disturbed soil. So you can also think about your deep soil as a bit of a bank where you're sort of preserving some microbes. So after the disturbance, they have something to grow back up into that disturbed soil.
Do you have a sense, Rick, on the rate in which that happens? Like after a disturbance event, how quickly are the communities able to rebound?
It's really variable among different kinds of microbes. And since I won't get through all of this, I'm gonna race to a certain slide that I like. This one. So I was talking about, this is like one of the lower topics anyway, the soil health practices. But minimizing disturbance. I like this. This isn't my research. This was done in Iowa. This is looking at arbuscular mycorrhizal fungi, right? So these are those beneficial fungal symbionts of herbaceous plants, so that all of your grain crops would be using these. And this is like, and NT is no-till, CT is chisel-till, I think. So just, one tillage pass, you really drop these guys. But what was interesting to me in the study is they did a follow-up. So they went back to these plots, 1, 2, and 5 years later. And this is a single tillage pass. So the difference between blue and orange there, they haven't kept tilling it. This was the tillage they did in year 0. Look, 5 years later, you're looking at the percent decline in these mycorrhizal fungi. And so you knock them back. This particular group of fungi, which are a pretty important one, they're your plants and plants, are really slow to come back. They're very sensitive to disturbance, but also they're terrible dispersers. Some bacteria and fungi fly through the air. So if you knock them out of the system, if they're in the area, they'll fly back in. These guys never go above ground. So they're not flyers. So if you knock them out of your system, it takes a long time for them to come back.
One thing, not to challenge, I know you have a ton of slides to go through, not to debate this, but I wonder if that system, if it's no-till conventional versus tilled conventional, includes cover crops, because I think that's really a really important factor too.
Yeah, I'm not going to remember the explicit details here. I know that those would have been passed after that one tillage event. These experimental plots would have been managed the same. right? So it's not like the blue bars are getting cover crops and the orange bars aren't. But I don't know what system they're being managed under. And it could be that, right, with a different rotation system, with cover crops, you could build that community back up faster, right?
I have a question, sorry. I don't entirely agree with the notion that stratification in agriculture is good, like what you said in the no-till systems, because it leads to zone-specific depletion of nutrients. And you alluded to it earlier when you were talking about straw versus clover material digestion. And I think what you said about that was a little bit of an oversimplification. It's really about the stoichiometric ratio. So to process 1 unit of carbon, microbes need a certain amount of N, a certain amount of P and S. So can you speak a little bit more to diversity of residue quality and why having nutrient balance is important?
Yeah, well, it's all about the goal, right? What is your goal, right? Putting carbon in the mineral-associated pool is only one goal. The issue and the challenge with soil management is that we're trying to maximize many goals at the same time. So any one strategy is going to be good for one goal and maybe not great for the other goal. So diversity of strategies, like you're saying, having a diversity of different inputs may get you better overall outcomes in terms of getting better outcomes throughout on multiple axes, if you will. The idea with litter quality, and carbon to nitrogen ratios are part of litter quality, they're not all of it, right? There are also differences in the carbon, the actual structure of the carbon, like lignin versus cellulose versus xylose, in terms of how microbes deal with them. But the sort of reigning, as you say, simplified paradigm is that it's about the carbon use efficiency of the microbes, right? For what kind of litter will the microbes use that carbon more efficiently in terms of translating unit of carbon in versus unit of carbon incorporated into microbial biomass versus unit of carbon respired as CO2. And so,
yeah, I guess my real point for this audience is that, like, with no-till, you can get nutrient stratification, and occasionally people see benefits with occasional mixing because you're getting nutrients to marry together in a way that benefits biology.
yeah, and I think that would be, I could see that argument. It'll probably depend on the crop that you're growing and its ability to scavenge nutrients from different soil horizons. If you're growing a crop that has if you're continuously growing a crop that has all of its root zones in all of its root in one zone in the soil, then you could deplete that zone of anything and have this big pool of nitrogen or phosphorus that's like just below those roots that never gets touched. And so then mixing them together puts the nutrients back into that zone where your roots are. On the other hand, if you grow a diversity of crops with a diversity of rooting strategies, that's maybe less important because you have a deeper rooted species that's going to get to those deeper nutrients, right? And the shallow rooted species, shallow nutrients. So there are probably multiple ways to deal with that problem of nutrient depletion.
Get one more question and let Rick get on the
Yeah.
slides.
Could you just explain the second slide?
Yeah, the second graph, sorry, it's a little confusing. What's on the y-axis there is a percent reduction. So if you compared the plot that had been tilled versus the plot that had not been tilled, how much higher is the mycorrhizal population? Is it, or how much lower is the microbial population in the tilled plot? Is it 50% of what you get in the no-till plot, or is it only, is it a 30% reduction or a 40% reduction? The point there is not zero. If the mycorrhizal population had completely recovered, those bars would be at zero. The blue and orange is just two different sites that they did this in. Sorry about that. And so the only point I was trying to get at that slide is that if you go across the years, those bars go lower, so indicating that there is some recovery happening, but it's not down to zero, and it took a long time. All right. Remind me again. How long do I have to, Aaron?
We built in some nice
Okay.
breaks to buffer.
All right,
So we don't have to cut
Yeah.
talk short. I'd say till 9:20, another
All right.
half hour or so.
Folks want to hear a little about the current research. That's good because it's quick, because I didn't put much in there. But I wanted to highlight this study that Aaron Greg is in the audience. Leia's here in the audience. You'll hear from all of these folks in a minute. So my lab was involved in this too, where we, if you guys are not familiar with the Wisconsin Integrated Cropping System Trial, you've probably heard about it before in these kind of venues. It's a wonderful thing that I take no credit for, because I was not involved in it, but totally took advantage of it to look at these long-term effects on soil microbial communities. And so just the idea here is it's got these different cropping systems that are different in many ways. Some of them are organically managed, some are conventionally managed, but they also differ in the rotation and the cover crops and all sorts of things. But they've been running since 1989. So it's one of the really rare opportunities where in a sort of experimental way, we can look at long-term consequences of different management decisions. And so just results here from this paper that, as I said, first author was a former postdoc at UW, but then there's Leia there, the second author, Greg's on there, Aaron's on there, so it's this team. Just two quick results. One is just ask, if we manage a system in a certain way for a long time, do you develop distinct microbial communities? And so you can see these graphs, the only thing to understand about these graphs, look at the colors. The 3 dots of each color would be replicate plots of that cropping system. And if the dots are close together, they have a more similar community of microbes. This is looking at which bacterial and which fungal species are there. And so each system, to a greater or lesser degree, develops some degree of uniqueness in its community. Not surprisingly, the continuous corn system has a fairly unique community compared to the pasture. Those are the big differences. Not surprising. A little easier to interpret are these differences in the biomass and the diversity of the microbial community. So I'd especially point you to that bottom one, and this goes back to that comment I made about, do I care about the ratio of fungal to bacteria? More, I want to know how much fungi, how much bacteria. And there you can see that these cropping systems were affecting both of those pools, both of those aspects of the soil community in similar ways. The continuous corn system had the lowest biomass of fungi and the lowest biomass of bacteria. The pasture had the highest biomass of both those things. So the ratio may not have been changing much, but the total pool is going up in that more diverse, less disturbed system. The forage systems, which have that, are sort of intermediate in both the diversity and the disturbance, are falling out in the middle there. So what do you do with this? Mostly this, for us, served as some kind of proof of concept that if we make big changes to a system, we can eventually see changes to the soil system, to the microbial component of the soil system. But you'll notice not a lot of distinct differences between really similar, cropping systems that on this scale are kind of similar to each other. In reality, I realize that that's a really different system to go from a continuous corn to an organic, corn, soybean, wheat rotation, right? Like that's a huge change from the management perspective. But from the microbial perspective, we didn't see a big change from that. So that's a little bit of sort of a sobering idea. All right. And then there was some interest in talking about the role of microbial products, commercial microbial products. So I have quite a bit here. And again, this is not, I don't have information about any individual microbial product, right? And I don't do a lot of product testing in my lab group. So I'm going to talk kind of more generally about some of the ideas behind this. So can we add diversity for microbial inoculant products, or can we add targeted microbes to achieve targeted outcomes? Lots of people want to do this for various reasons. This is becoming a pretty big business, right? The quote up there from, this is old and out of date now, but Hugh Grant, or at least at the time, was a big leader at Monsanto. It's all about microbial products as the next big thing in the ag tech industry. Part of the motivation, honestly, is that the regulation to bring a microbial product on the market is almost nil compared to the regulation involved in bringing a new GMO on the market, which is decades and millions of dollars to do. So this is an easier path for the biotech industry. And so, yeah, people are buying them. So, right, another poll for you guys. Yes, vote. All right, I won't belabor this. Just to get the idea, at least some folks are using these. That's not so far the majority. So I'm going to cut you guys off and go to the next question. So refresh the website to get a chance to enter a new answer. If you said yes, you can answer this. Or even if you said no, if you are thinking about it, if you're curious, if you're microbial product curious, you can answer here, like, what would you want to try? Okay. That's interesting to me. Let's go back and talk a little bit about my thoughts. And some of this I will now preface is a little bit my opinion. I like to think it's somewhat informed, but it's also obviously open to disagreement. Because there's one of the unfortunate things about this space is that I don't have a lot of really hard data to show you on it. I should do that. So just a little background. When I talk about microbial pesticides or biocontrol agents, that's something, a live organism that you're putting in to your soil or onto your plants with the goal of trying to control one or more pathogens. There are a lot of these on the market. They're used in conventional systems, so they're also used in organic systems, but also used some in conventional systems. Probably the microbial products we best understand. Then we have these more general microbial soil treatments. I see these much more often used among organic growers than any conventional growers that I know about. And these are where you're trying to increase plant growth through some kind of effect on nutrient acquisition or fertility. That was what I was calling biofertilizers before. Or affect plant growth in some much more mysterious way, the ones that are just called plant growth promoters. How is that happening? What's the magic there? To the extent that we understand that magic, it's often about hormonal control of plants. And so it's better to think of these as plant growth manipulators than as plant growth promoters, because they're manipulating plant growth by producing hormonal mimics. Okay, so the big question that I'm often asked, if I buy this thing and I put it in my soil, will it work? And so when is it going to be possible to successfully add a new microbial species to your community? And so that's one thing that has to be true. And the second thing is that it has to then achieve some additional beneficial function as a result. So again, I'm not going to present like a list of products and ones that work and don't work. This is more just a thought process for something that you could think about if you were considering trying something. So first I thought I'd start with some sobering math, just like how much is in your soil to begin with, and then how much are you adding. So if we take this really rough, somewhat made up number from an RCS, take the low end and say we have 100 million cells, bacterial cells in a teaspoon of soil, which is about a milliliter. And scale that up to 1 acre of a field. Go to a depth of 20 centimeters. So you think about that volume of soil, right? A teaspoon, now we're going to like an acre. So I did a little bit of math yesterday. That translates to around 16 quadrillion bacterial cells in an acre of soil. All right, so 1.6 times 10 to the 15 cells per acre. So then I went and I just picked, like completely at random, top Google hit of one of those microbial soil amendments. So not a biocontrol product, but this was marketed as a nutrient efficiency optimizer. I'm not sure what that means. But it was a methylobacterium product. So the methylobacterium is a common soil microbe. And I like this one because the label actually gave me some information about it. 3 times 10 to the 7th cells per gram. They suggest you add 5 ounces per acre. So that comes out to 4.26 billion cells you're adding to your field, which seems like a lot. 4 billion cells you're adding with this product. But just want to point out that the acre already had 16 quadrillion cells in it, right? So if you work that out, your inoculated cell, you're adding like 1 inoculated microbial cell for every 400,000 indigenous cells. In this like back of the envelope math, obviously the numbers will change. I bring that up just so you keep in mind that that cell better be pretty special if it's going to do something that those other 400,000 cells are not doing. Which is not to say that it can't, but that should be sort of your baseline starting point, right? It's got to be pretty special. The other thing I want to emphasize about microbial products, everything about biology, everything about ecology, and soils are a very complex ecosystem, everything is context dependent. So even something like mycorrhizal fungus is our textbook example of a beneficial microbe. But that's not always true. It's a nutrient exchange relationship. It costs the plant has to pay the fungus in exchange for the nutrients the fungus is providing. So if the nutrients are in our excess in the soil, that fungus is a drain on the plant. So mycorrhizal fungi can be parasitic in certain conditions. So just keep that in mind. Just because a product worked on your neighbor's field doesn't mean it's going to work on your field. Any questions, Mike?
Often an input doesn't work because your field didn't need it, not because it's a bad input.
Yes. Well, yes, that's an argument. So like to say a mycorrhizal fungus is parasitic is, you can argue that's not the right word. It's doing what it always tries to do, which is provide nutrients to the plant, but it demands carbon in return. If your plant doesn't need those nutrients, the net outcome is growth reduction for the plant. It's not like the fungus became a pathogen or anything. All right, so these are the questions I would ask if I were considering buying a product, right? So one, as was brought up, does my crop need this organism? The function that it's supposed to be providing, is that a function that I need in my soil? And so it's kind of in a hierarchy. Like if the answer is yes, then you go to the next question. If yes, I do think I need this function, is this organism already in my soil? Soils have a lot of stuff in them. Maybe you already have it. If you don't have it, is some other organism, you have some other organism that's already doing the job, right? It's not like there's only one microbe in the world that can do most of these functions. And then even if all of these things are true, it still has to work in your soil, right? So if you add it, is it going to survive in whatever way you're adding it? And if it can survive, can it actually sort of compete and grow its population in the community that you already have? And so I'll just walk through these a little bit. Again, I don't have answers, but these are questions you can ask, right? So even in my quick survey of sort of what products are out there, some of the products do a good job of saying, this is what this is for, right? This is for the control of this particular pathogen. This is a microbe that fixes nitrogen from the atmosphere and could provide it to your crops. But other products are much vaguer, like put this in the soil and your plants will be healthier. So I would be skeptical of that. Like if they can't tell you why it's working, then it's going to be really hard to answer this question. Do I need it? Well, I don't even know what it's doing. So yeah. And then, is there some organism already in my soil doing this? That's really hard to know, right? Because you don't know what microbes you have in your soil. None of the soil health tests that are available are going to give you that level of specificity, right? They might tell you the total amount of microbes you have in your soil, maybe how many fungi you have versus bacteria, but again, there can be hundreds of thousands of different bacterial species and fungal species. So how are you going to know if this particular fungus or bacteria is not already in your soil? There is a way to know that. In our research labs, we answer this question all the time. There are, I think there are some commercial services now that will do the kind of microbial community screening that we do in our research program, do it commercially. But what we lack is sort of the interpretation of data. It's becoming easier to get that data, but of those thousands of microbes that you find, most of them we don't know really what they do. And so that service isn't going to be able to give you a confident answer of like, oh, because you have these microbes, you're going to have these problems or whatever. The science isn't there yet, I would say. But I can speak a little bit, again, trying to come up with some sort of, at least first principles you can use to guide your decision making. those microbial functions, like what do you want that microbe to do for you? Microbial functions run this gradient of being what I call generalized or specialized. So a generalized function is something that a lot of different microbes can do. So things like decomposition, that mineralization of nitrogen, so like freeing up nitrogen from your organic matter, contributing to soil aggregation. These are broad-scale functions that lots of microbes contribute to. It's not like there's one microbe that can degrade corn leaves, and there's a different microbe that can degrade soybean leaves. This is broadly distributed. So, and then specialized functions are something where only a really narrow set of microbial species can do that. So some of those, suppression of a specific pathogen, that might come down to a very specific molecular mechanism, production of some antibiotic that this inhibitor, this antagonist microbe can do that most other microbes can't. The production of those plant growth manipulating hormones, for instance. And that's a relatively narrow function. So if you're worried about general function, much more likely that you already have microbes in your soil doing it, because most microbes do it. What you would probably, in my opinion, be better focused on is like building up the biomass of that general microbial community in your soil. Because as you build up more microbes, they're going to keep doing these general things. So these are the processes that you're getting the best information on from those soil health indicators that are available. So you can focus on that. It's also really unlikely that these processes would be materially affected by adding 1 or 2 inoculated products, right? Just go back to that math of like how many cells are you adding versus how many cells are already there. This is a general phenomenon that most of the microbes are doing, then you're just adding a tiny bit to an ocean of microbes. On the other hand, you can have more specialized functions. In this case, those soil health metrics are not giving you information about these specialized functions. So you don't know. But it's also more likely that this function is going to depend on the presence of a specific microbial species. And so maybe you don't have that species. Maybe adding it would be a good idea. So again, it's hard to know whether you have the species in your soil or not, but if you're considering buying the product because you have a problem you're trying to solve, that may be one indication that your current soil system is not solving that problem. So maybe it's worth trying something. These last 2 questions, again, are really hard to answer because of that context dependency. It's not something that a producer, like a company, will be able to tell you because this answer depends on your soil, right? But you can ask, has it been tested on your soil type, right? On your crop species, on soils like your soil. If it's only been tested on soils that are really different from your soil, then you should be skeptical about whether this is going to work. And I think most, by the time they're selling it to you, I hope this is true, but not always. Has the product been shown not just to survive, but to show efficacy in natural, soil conditions and not in sort of laboratory studies? It's really easy to find a microbe that does something on a petri dish. Much more difficult to find a microbe that will replicate that process in soil, for all the reasons that we've just discussed. So I hope that didn't come out too nihilistic or skeptical. I think there is a possibility that a microbial product could do something good, but there are a lot of reasons why it won't, I guess is what I'll say. And unfortunately, it's hard to know before you try it whether that's going to be the case, but I hope these questions help you at least think through, is this going to be a high probability of getting a good outcome? And you can balance that against the cost, right? What's it going to take for you to try this? As a researcher, my response is always like, well, you need to test it. You need to test it. You do an experiment. That's what I do. So I do encourage, if that's something that you think you can pull off, that you treat a portion of your field, right, and see how it goes.
I just can't stress that enough, Rick. And like you said, these are ecological, biological systems, and which makes them exciting to work in, which makes an opportunity for a lot of potential. But with any new practice or product, to do on-farm experiments, and I get frustrated with the lack of data coming out of our university systems with these product tests, but honestly, I also question, I can test these at Arlington, but does the data that I would produce, which I haven't done a lot of testing, but would it even be relevant? So I think that if you're thinking of this, to do these controls, there's actually some great resources with respect to doing on-farm data collection. Doesn't have to get overly complicated. Now, PFI is amazing with their networks, and I know Doug and Allert and Margaret Smith have been really involved with some of those trials in the past, but put it in a section of the field that's representative of the field. Try to do replication. Don't just put out one small strip. Make sure you have a control strip. And I get really excited when I try new things, and what we think we see is not always what's out there. And I know my research scientists, Leah and Ben, as I get excited, I'm like, oh my gosh, it worked, it works. And they're like, wait, let's collect the data. It might not be what you're seeing. So I think we all just as humans have a natural tendency to get excited and our eyes tend to see what we want to see, but actually getting the data, doing these strip tests, doing replications, and actually doing it over multiple years. So unfortunately in agriculture, we have one shot every year to collect these data and try these things, and it's frustrating the time it takes. I love the work that PFI is doing and these on-farm research networks so that we can collect information on farms to make data-driven decisions, to be able to calculate ROI, to be able to start to understand where do these work, when do these work, how many years do these work, and I hope we can start expanding that through O-grain as well, but I just can't emphasize, the lack of data I think is frustrating. I agree, Rick, I'm a skeptic. That's not to say I think there's not potential here, but I just get nervous that about, and I don't, one thing I love about organic is it's not an input-driven system, and how, if we're going to smartly use impact or inputs that are going to be beneficial to the longer-term ecosystem as well as beneficial to our pocketbooks, just be strategic and smart about what inputs we're choosing.
Yeah, it goes in, so
I just have a comment, on the soil health and in the beginning of your presentation. I had one topic that, again, I don't want to open a can of worms or do a debate, but soil health and its impact on groundwater. My county here, Portage County, 90 miles to the north, we've got an enormous amount of large-scale vegetable growers, and we've got people that, point fingers, where is it coming from? We've got water quality issues, high nitrates. A challenge to the university to do the us in this room are organic farmers. We feel good about our soil. We feel good about the groundwater that we're going to pass on to our children. But what about the other guys? Do they have access to this data? Do they have access to the, they'll come through, they'll fumigate, they'll kill everything through, sand will blow through, so.
yeah, I will say I personally work quite a bit with large-scale potato growers in the Central Sands.
Okay.
Oh my God, absolutely. Everyone is very concerned about nitrate in the water. And it's challenging. If you grow there, you know the challenges, right?
Yeah.
Sandy soils, everything just flows right through them. A lot of this stuff we're talking about, like build up your aggregations. I've never seen an aggregate in the Central Sands. It doesn't matter how much or little you till. It's just not that kind of soil, right? And so that is another pet peeve I have a little bit about when we talk about soil health. It's so broad in those 4 building practices or whatever. Like, you got to work with what you have, right? And same with the carbon sequestration thing. Like, not all soils are going to sequester large amounts of carbon. It depends on your history of glaciation, right? And the bedrock material you're working with. We can't expect everyone to get to the same outcome. And so farming on sand is super tough. And a lot of this stuff is maybe not as relevant. It's inherently a more input-output system just because the soils don't hold on to things. And so you've got to deal with that, and it's more about, being really precise about when I drip the nitrate in so that it's meeting the crop demand and not, because if there's any excess nitrate in the soil, it's going to be gone in a week, right, into the groundwater, which is really different in a loam soil. So, I don't know. I don't have an answer for you. I know everyone's thinking about it.
Carmen has a last time. Oh, Jared, go ahead. Okay.
I go first.
Time
I was
and a half
first.
under control.
I need a simple answer to a complex question.
Awesome. I'm great at that.
Driving back to organic matter and measuring that. So I appreciate the breakdown of kind of the two types. That's the first I've heard this. But for the redneck simpletons like me, we take the sample, we mail it in, and a number comes back, and it might be 1 point, 2 point, 3 point. Now I also understand that that's Logan Labs and Midwest Labs. And then there's also the soil test, the Haney soil test. But the problem with that one is you take your best field, your most productive field and that gets a really crappy overall health score, but then you take some miserable piece of like pasture land. And so for the rednecks who just want to take a soil sample, get a number, if the number goes up, things are probably trending right. If it's going down, you're doing something wrong.
Right.
Is it worth the money for that extra Haney test?
The Haney
or
test.
are we good with the so just a
Yeah.
simple answer for a complex question.
Sure, right? And the Haney test, like you can get more complex than the Haney test. You can go to the Cornell, the Cash test does more things. There are other outfits out there testing all those other things. Yeah, I don't know. I don't have a simple answer for you. Those extra value-added things you're getting from the Haney test, like that soil respiration burst, is one thing they do. It's supposed to measure microbial activity. Some of the other ones do these like the active carbon pool, protein, the ACE protein, which is getting at organic nitrogen. The good thing about those, I guess, if I make a positive argument for them, is that that basic organic matter percentage you're talking about, that loss of organic matter, that is not going to change fast, no matter what you do. Like, that is you can lose it, for sure, over decades. Building it is super hard. I mean, Greg's here, right? I don't know if we'll talk about the Wicks trial. Like, they were comparing best practices to worst practices for 30 years, right? And everything lost carbon. So the hope of those expensive tests is that they're more responsive. So if you want to know if the thing you're doing is having any impact on your soil, you're more likely to see it in those active carbon test and that soil respiration test. Downside is, I don't really know what to make of the numbers. Do you want it to be higher? Is there some optimal number you should be aiming for? Is there a number that's too high? I don't know. So they tell you whether what you're doing is making a change, but I'm not convinced that we know what change you want to make, right?
A question I get 100 times a day and over the years is, do herbicides impact soil bacteria and soil fungus? And the other piece would be, what is the impact of varying weather conditions on the fungus and the bacteria?
Yeah, well, the first one, I'll be careful about going on a limb without knowing the data stone cold. I'm sure it depends a bit on the herbicide. The big one that we use most, glyphosate. Good news about that one is that it is very targeted to a very plant-specific mechanism, right? It inhibits the photosynthesis. So many people have looked and not found much evidence of an effect on microbial communities in soils directly. I mean, as opposed to like having weeds in your system probably does affect your microbe community because microbes respond to plants. And so if you get rid of the weeds with glyphosate, then it's going to change for that reason. But the chemical itself. Now, I don't know the data on like atrazine or dicambra or 2,4-D, so I don't want to say something out of turn. I'm not aware of any clear studies that document a big change with those. The weather thing, microbes are really responsive to soil moisture, desiccation. But when your soils dry out, that's probably the biggest thing that affects microbes. Some microbes are good at sort of tolerating that, like going into some dormant state and then waking up when the soil gets wet again. one thing I do find interesting is that ability of microbes to sort of tolerate dryness, dry spells, and then wake up when it's wet, and then go to sleep again when it's dry. That is a trait that they will evolve if they've had a history of those wet and dry cycles. So if you're say somewhere in California, like a Mediterranean climate, where you have extremely hot and very dry summers, those soils, I used to live out there, the soils get baked, and then it rains in the winter, and then everything wakes up. Microbes out there have evolved for millennia to respond to that. Up here in the Midwest, we don't get that same kind of, or at least historically haven't gotten that same kind of hot, dry summer that really bakes your soil. So the microbes out here, my suspicion is that they are going to be less adapted to those extreme wet and dry cycles. It also makes me question, going back to the question about the Haney test. They do that microbial respiration. They take your soil, they dry it completely, and then they wet it again and then see how much respiration there is. I'm like, that's a terrible idea for Midwestern microbes. I think in that drying step, you've probably killed a ton of them, and they're not coming back. So question of how relevant that is, to actually predicting microbial activity in our fields.